Hello, everyone. Thank you so much for joining our webinar today. I'm excited to have Jamie Williams and Lily Zafis here with me today hosting the webinar. I'm Katie Breinig, and I will be moderating today's session. Before we begin, I would like to let you know a little bit more about how this webinar will run. This webinar is a two-part series. Part one, which you are attending today, is on off-flavor and off-odor. And we will be hosting part 2 foreign materials and unknown objects on August 17th from 11 a.m. to 12 p.m. Central Time. The link to register is located in the chat below. Today's webinar is being recorded, and the slides and the recording will be available for you within 3 business days. A short Q&A will follow the presentation to answer viewer-submitted questions. During this webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your questions, and then hit Enter. Remember, you can submit questions throughout the webinar. Jamie, I will now pass this webinar off to you. You may begin your presentation.
Well, thank you, Katie, for that introduction. As mentioned, I'm Dr. Jamie Williams, and I work as a senior scientist at Eurofins SF Analytical. Today, along with my coworker Lily, we're going to be presenting on GCMS techniques for troubleshooting of off odors, off flavors, and flavor scalping in food, beverages, and related products. So just a general overview of what we're going to be presenting on today. First will be an introduction to off odors and off flavors and common sources and causes of these issues. Following this, I'm going to talk a bit about the background of the analytical technique, that is the actual scientific approach we use. And then I'm going to hand it off to Lily, who's going to talk about some case studies to give you an overview of how this approach can be applied to real-world problems. So, as I'm sure you're all aware, aroma and taste are important components of consumer appeal. And taken together, they constitute the flavor of a product. And a product's flavor has many important influences, such as being an important part of a product's identity. As well, changing or modifying a flavor is important to new product formulations, to stand out from previous products, or to stand out from a competitor. As well, the flavor of a product can be important in quality assurance, as off odors and off flavors can be indicative of issues with a product. And finally, flavor is an important part of consumer trust. Consumers want to know what they will be purchasing and that they can get the same consistent product every time. And so off odors and off flavors are not only off-putting to consumers, but they may be signs of serious problems. And very broadly, off odors and off flavors can be broken down, or causes of off odors and off flavors can be broken down into 3 major groups. These include packaging issues, chemical or microbial contamination, and production issues. And there's a fair amount of overlap between these classes, as you will see. So when we're talking about packaging issues, this can result in a number of different problems. The first of these is packaging incompatibility, where you have selected a packaging material for your product that isn't appropriate. This can include using packaging that reacts with the product or using a non-food grade material. And packaging incompatibilities can lead to flavor scalping. And in flavor scalping, components from the product are absorbed into the packaging material. And this can lead to a loss of flavor and odor compounds over time, resulting in a decrease in product quality during storage. An example of scalping, a visual example of scalping that you might have seen yourself, is if you've ever had a tomato-based product in a plastic container, especially if you heat it, you might find some of those color molecules move into the container from the product. In reverse, we can also have leaching from packaging material. And this is where components from the packaging move into the product. And this can be a potential safety concern. A common example of this is the issues with BPA leaching from plastics into products. Also, another example of this would be back when canning was first introduced. A lot of the cans that held acidic products would degrade over time due to the acidity, resulting in a metallic flavor to a product. Related, we can also get product degradation if an incompatible packaging material is selected. This can occur if you're storing a product that has light-sensitive components in a clear packaging material. And so over time, exposure to light can lead to oxidation of components, which can lead to breakdown of some of the flavor compounds, but can also lead to oxidation of compounds such as unsaturated fatty acids, leading to rancid or stale flavors. Relatedly, if a product is stored in, for example, a gas-permeable packaging when it should be stored under an inert gas, you can also get oxidation due to the presence of oxygen. Another common packaging issue would be just using packaging that isn't sturdy enough or isn't sealing properly. And there you can get the packaging to start to break down, or you might get some damage to the packaging, which allows material to leach into the product. And this can lead to contamination. And so contamination is another common source of off odors and off flavors. And this can also be broadly broken down into two classes: chemical and microbial contamination. So when you talk about chemical contamination, there's a wide variety of different sources. This can be contamination with potentially hazardous materials, cleaning agents, heavy metals, and the like, as well as non-hazardous but unwanted materials such as cross-contamination from another food product. And so chemical contamination can arise from storage issues, such as leaching from a storage receptacle, leaching from the packaging, or just improper storage, like I mentioned before. A damage to the packaging material can allow unwanted chemicals into a product. Improper cleaning is a very common source of chemical contamination that can lead to off odors and off flavors. One of these would be, for example, insufficient rinsing procedures, and then you might get cross-contamination between an old product and a new product. And so now your new product maybe has a bit of the flavor of the previous product that was used on a production line. Something else that is very common is contamination with cleaning agents, where a production line or a packaging facility was washed with a cleaning agent and that wasn't thoroughly rinsed out of the system. In that case, you can get contamination in your product, which often leads to solvent or chemical-like off odors and off flavors, or sometimes acidic off flavors if the cleaner was acidic. And finally, user error is another common source of chemical contamination. This can happen during production, during storage and shipping, as well as in a customer's home. An example of this would be excessive lubricant added to machinery, which then contaminates the product. As well, this could be as simple as someone leaving the cap off a container and accidentally contaminating it from the environment, perhaps even a customer in their own home, and then they taste an off flavor and want to know what is wrong. Microbial contamination can arise from very similar sources as well. Once again, improper storage. For example, storing a product that should be kept frozen at room temperature too long can allow microbial growth over time. Improper handling, if improper cleaning or cleaning isn't being done properly, you can get contamination in that way. Maybe people aren't washing their hands or wearing their proper equipment, such as gloves. You can get contamination with microbes. Cross-contamination between a product that's supposed to be cooked, such as a meat, with a product that won't be cooked. And finally, process failure, such as incomplete pasteurization, can lead to long-term microbial growth. And microbial growth can lead to a variety of off odors and off flavors, most commonly fermented or acidic off odors and off flavors. And it can also lead to other effects on quality, such as bloating of packaging due to the buildup of gases. Finally, we have production issues. So here, this can be broken down as well. For example, an equipment failure can lead to an off odor, off flavor if it isn't caught and rectified. So for example, if an oven isn't reaching the proper temperature and no one notices, you can get incomplete cooking of a product. This can lead back to microbial contamination, but can also lead to other off odors and off flavors. For example, say you have a baked good and it's not being baked long enough, so you're not developing that nice browning and crust that is typically associated with something like, say, a bread. So you're not getting the desired flavor profile. Related would be a process failure. This would be where a process isn't designed properly, and so it can be related to the equipment, but in this case, it's not an issue with the equipment, but rather the design of the process itself. So for example, if a process doesn't have pasteurization leading long enough, this can lead to, once again, microbial growth. Or another example of this is if a product that needs to be defatted isn't properly fully defatted. This can lead to an excess of fatty acids in the material, which can long-term result in oxidation, in rancid or stale-like flavors. Once again, user error comes up, and this would be, for example, failure to recognize an calibration equipment. However, this can also be a wide variety of other issues. For example, you could have a user accidentally add twice as much of an ingredient as they were supposed to, or perhaps forget an ingredient. Now your flavor profile is off. This can also relate back to contamination. Say, for example, someone accidentally drops their cell phone in a vat of material. Now all of a sudden you have concerns about contamination from that cell phone. And finally, ingredient issues. This can come in a variety of forms, such as impure ingredients, resulting in off flavors due to the impurities in the ingredient itself, as well as changes in quality of ingredients with a new supplier. In some cases, this might not even be a decrease in quality, just a change in the overall profile. But you might find that over time, your product starts to taste a little different. Maybe it's not an off flavor, but just a change in the profile. And this is common with natural ingredients, as every fruit and vegetable is a little bit different. And in many cases, an off odor or off flavor is caused by a combination of system and human errors. So there are a lot of different potential causes of off odors and off flavors. So how do you go about narrowing that down? Now you have a product, your quality assurance comes to you and they say, this doesn't taste quite right, or perhaps you've gotten a complaint from a customer. Where do you even begin to look? Well, we use an approach known as aromatic, or sometimes volatile profiling. And so what we do is we look at the profile of volatile compounds in a product. And it is this profile that contributes to the overall aroma of a product. So for example, on this slide, I have the profile of a coffee type product. And each of the peaks you see in this image corresponds to a different chemical compound that's part of the volatile profile. And below I have a table where we've identified a number of these. And as you can see, a lot of them give nutty and roasted aromas, which you would expect for a coffee-based product. However, we're also seeing green and sour and fruity aromas, which help contribute to the overall body of the product. And so it's not just one or two compounds that contribute to the flavor and aroma of a product, but a wide variety of different ones taken together in their different relative amounts. And so how do we go about identifying compounds that might be responsible for an off odor when these profiles tend to be quite complex? Well, we do this by looking at a control sample and the sample that has the off odor, or the suspect sample. Ideally, these two products are as similar as possible. So for example, different batches of the same product, where the good batch doesn't have the off odor and the bad batch does. And then we can look at these profiles and look for differences between the control and the suspect. So for example, say we're analyzing a control and a suspect, and we see in the suspect product a peak that we don't observe in the control. From there, we can look further and maybe we identify it as a solvent or a fragrance related to a cleaning agent. And then you can trace back the off flavor and off odor to a cleaning issue. Likewise, we can see differences in the other direction. Maybe your control product has peaks that aren't in your suspect. So for example, say your control product has a peak for vanillin, which we don't observe in the suspect. Well, in that case, maybe vanilla was forgotten to be added to the suspect sample, and now your flavor profile is off. And so in this way, we can investigate the source of an off odor. And so it's all well and good to talk about looking at volatile profiles, but how do we actually determine those for a product? And we use a technique known as gas chromatography coupled with mass spectrometry, or GCMS for short. And GCMS is a powerful technique and our flagship approach for the analysis of volatile profiles. In this approach, compounds are volatilized and separated in a gas phase based on their affinity to a stationary phase. And we can tailor this approach to different classes of compounds by changing our stationary phase and our temperature gradient that is used during separation. Once separated, these compounds are introduced to a mass spectrometer, and this is used not only for detection, but also identification. As in the mass spec, these compounds are fragmented into small pieces, and the pattern of these fragments are characteristic to individual chemical components. So by looking at these characteristic fragmentation patterns, we can identify compounds by comparison to our libraries of over 300,000 compounds. So just to give you a visual overview of this process, first, samples are introduced into our GC system. From there, any volatile compounds are separated on the GC column, or stationary phase, with a temperature gradient. From there, they are introduced into the mass spec and broken down into smaller pieces. These compounds are then sent through a quadrupole in order to separate them based on the mass of these fragments, and then to the detector. And from there, we can get our profile and our fingerprint fragmentation patterns. So just to give you one more example of what this looks like, on the slide, I have the volatile profile of an essential oil. So a product that is known for having a very strong aromatic profile. As you can see, there are a lot of peaks and a lot of compounds present in this sample. So if we want to look specifically at just one, this peak here, we can look further and look at its fingerprint fragmentation pattern and compare that pattern to our library of compounds. And by doing so, we can identify this compound as 2-boronone, which has kind of an evergreen or kind of green-like aroma. And so in that way, we can look at the volatile profiles of our samples. But how do we get the volatile compounds from the sample into the GC? And there are a number of different ways we can do our sampling, with our most powerful technique being known as solid phase microextraction, or SPME for short. And SPME is a technique for the selective extraction of volatile compounds from a sample. SPME works by using a fiber coated with a thin layer of polymer material. And this material acts as an extracting phase, allowing for the selective extraction and concentration of volatile compounds. This is beneficial as it increases our sensitivity and selectivity. That is, we can use these fibers to concentrate the volatile compounds, reducing our detection limits, as well as we can select fibers based on the class of compounds of interest in order to target this approach. And in this way, we can work with a variety of different matrices without having to worry about the matrix effects that these samples can have. That is, food and related products tend to have very complex matrices that can interfere with certain analyses. And in this way, we can kind of sidestep some of those issues, and it allows us to analyze even very complex materials. So in brief, for SPME, samples are incubated in gas-tight Headspace vials in order to release volatile compounds. And by tailoring the time and temperature of this incubation, we can target towards more volatile compounds using lower temperatures, or less volatile compounds using higher temperatures and longer times. After incubation, the fiber is exposed to the generated headspace gas and allowed to equilibrate. During this time, compounds from the headspace gas are absorbed onto the fiber. Following this, any absorbed compounds are then thermally desorbed into the GC inlet and analyzed by GCMS as described earlier. In addition to SPME, there's a variety of other sampling techniques we can employ, depending upon the specific product of interest. One of these is Headspace-GC-MS, which is similar to SPME, but without the use of a fiber. So for some highly aromatic products, the concentration effect of SPME may not be desired, in that there may be so many volatile compounds present that it can overwhelm this approach. And so in this case, an aliquot of the unconcentrated headspace gas is used. Related, there are other approaches we can take as well if needed, such as solvent extraction and direct injection for less volatile compounds. So that was just a brief overview of off-odors in GCMS, and I'm going to turn you over to Lily for some case studies.
Awesome. Well, thank you very much, Jamie. So as she said, we're now going to dive into some case studies. So I'm going to show you what GCMS data looks like for real-world off-odor samples, and then we will show you how we interpret what we see and link it back to an actual product issue. So for our first case study, a consumer discovered an oddly colored fruity drink that also had an unusual aroma, and it made them question if it had possibly been contaminated. So this drink was usually bright red and fruity, and the product bottle that the consumer had found was a dark purple color, and it also had a chemical-like odor. So this is our first set of actual sample chromatograms here that I'm showing, and we have a control sample which had a normal appearance and aroma on the bottom in black, and then our suspect possibly contaminated sample was on top in pink. So when we analyze these samples, we're generating a huge amount of data. As you can see here, and as Jamie described, we have the chromatogram, and then each of these peaks that you can see is also identified by the mass spec fragmentation. And in addition to that, we can compare intensities of the peaks, which is related to peak height and peak area. So if a peak that is wide and tall, such as the one here at around 16 minutes, compared to a peak that is very small, that would mean that likely you have a lot more of that compound present. However, these intensities are relative, so we wouldn't compare between compounds because different compounds might have different responses even when they're present at the same amount, but we can compare between samples. So I'm going to highlight here the first differences that I want to point out. And in this case, we saw some obvious differences from the start. We saw the presence of isopropyl alcohol, 4,1-methylpropylphenol, and 2,4-dichlorophenol. And these are showing up in our suspect sample and not our control. So these compounds are all solvents that are associated with cleaning products and disinfecting agents, and this strongly supports that the sample was contaminated because we would not expect these compounds to be present naturally in this type of product, and because obviously we did not see them in our control. So in this case, we pursued additional analysis as well. Based on our GCMS results, we wanted to gain more information about the potential contaminating substance. And in this case, since we had a distinct difference in color, we also analyzed the samples using an internally developed and validated liquid chromatography method to screen for the major FD&C approved dyes. So these are the common dyes that are approved for use in food, drugs, and cosmetics, such as Blue 1, Red 40, all of the normal dyes that you might see on a food label. So this is a liquid chromatogram for the control sample. As you can see, it looks quite similar to our gas chromatography chromatograms. And what we identified here was the presence of Red 40 in the control sample. And this was expected based on the color of the sample and the ingredient list. And our suspect sample here, we also identified Red 40, but then we also identified Blue 1, which obviously should not have been there. So using this information, we were able to provide the client with the confirmation that yes, their product had been contaminated, and that if they wanted to further identify the actual contaminant, they should investigate for products that contain things like isopropyl alcohol and also Blue 1 dye. So our second case study, we had manufacturers of a newly developed fermented dairy product who noticed that some of their samples had a foamy surface and also an unpleasant off odor. So once again, we have our suspect on top in pink and our control on the bottom in black. And you might notice immediately that though the peaks generally are lined up with each other, there are significant differences in how intense these peaks are between the samples. So to highlight a few here, we're seeing significant differences with ethanol, acetoin, acetic acid, and 2,3-butane diol, most of which are more abundant in the suspect than the control. So in this case, elevation of these compounds, in particular ethanol, and also the foam that was observed, which is carbon dioxide, these indicate over-fermenting of the product, which could occur by multiple pathways. So in a fermented product, yeast are undergoing chemical reactions to produce different compounds. And in this case, there are multiple reactions that are likely occurring. As we saw in the control sample, 2,3-butanediol is a normal byproduct of these reactions, but in the case of our suspect sample, they were clearly having an over-fermenting issue, which was explaining both the off-odor and the foam that was observed. So our next case study, we had a freshly manufactured batch of vanilla smoothie, which was reported to have an uncharacteristic smell, which was described as solvent-like in some cases. Once again, our suspect is in pink on top and our control is in black. And you can see here, we once again have quite similar profiles, but there are some slight differences in intensities here in these compounds that I have highlighted. And when we dug further into this data, what we found was that these compounds that were less abundant in the suspect, which were vanillin, ethyl vanillin, and piperonal, these are all the major volatile components of vanilla. And as I said, they were less abundant in the suspect. And then in addition to that, the suspect actually had higher intensities of two compounds that are industrial precursors during the synthesis of vanilla, which were 2-ethoxyphenol and guaiacol. So in this case, this is an ingredient issue where the vanilla that was used in this bad batch was either degraded or potentially it was not properly produced, which led it to not be fully reacted. So we were able to inform the client and they could remediate their issue by getting some fresh vanilla extract. Our next study, we had a customer who was complaining about a powerful floral odor in their table sugar when they opened it. So once again, we have our suspect in pink and our control in black. And I want to point out also in this particular situation, our control sample was a retained sample. So the manufacturer was able to actually send us sugar of the exact same lot that the consumer had reported the off odor in, which is important. So as you can see, the control has almost no profile. This is what we would expect to see for a sample that doesn't have a strong aroma like a sugar. And there are many peaks that are very abundant in our suspects, so we have a lot of things to investigate right offhand. And I'm just going to identify several here. All of these in orange are solvent compounds. And then in blue, these are all associated with very strong floral or fruity aromas. So as I said, we have an abundance of excessive compounds that were found in our suspect sugar that were not found in the control. And these could be found in a variety of products, in particular cosmetics, soaps, and detergents. And based on the high levels of solvents, in addition to fragrance agents, it's likely a soap or detergent. So we do have a contamination, and in this case, as I had said, since the consumer and the retained sample that we compared were of the same lot, the manufacturer was able to contact the consumer and inform them of what they had found, and they also found that the consumer realized they had been storing this sample of sugar next to some strong detergent samples. So it was actually a consumer error issue, and we were able to confirm that using our techniques here. For our next study, we were looking at coconut flour. So this was an unflavored coconut flour powder, and it was reported to smell funny compared to a normal sample. So this was during the product development stage. So our client realized they were testing packaging, and all of a sudden they had this potential funny sample smell. So they sent us fresh control sample along with packaged suspect. And since the client was concerned about packaging, we also analyzed packaging material as a sample. So we still have our suspect here in pink and our control in black. And as I said, our packaging was included as a sample, so the blue profile is our actual packaging. And I want to point out a couple of things here. So these are just compounds that I'm pointing out. These are unique to the packaging. You can't see many compounds like this in this range of the chromatogram since I was highlighting other things, but it did have its own profile. And I also want to point out, I don't have arrows for it, but at around 12 minutes, you see there's a peak that is in all of the profiles. And you see this, that there is quite a bit of other overlap. And I wanted to note that these compounds are called siloxanes. These are artifacts from interaction with the column and with the fiber, so these are not actually real volatiles in the sample. So I just wanted to explain that as I move forward, that these are not actually for comparison. So what I do want to highlight, we have this compound that's showing up abundantly in the control, and then it is less abundant in the suspect, and it is also showing up in our packaging material. So as Jamie described earlier, this is a case of flavor scalping, where we are seeing the packaging absorb some of the volatiles from the sample, which leaves less in the sample itself. And then in addition, at the end of the chromatogram here, we see abundant peaks that are in the packaging, and we're actually seeing them show up in the suspect sample. So we also have some cases of leaching that are occurring here. So these results show that there's likely a complex cause of off-odor in this case, where we have both flavor scalping and leaching that are happening. So this is likely a packaging issue. Potentially, it just isn't compatible with their products because it's clearly both contributing volatiles and removing volatiles, which is leading to that funny aroma. So our next case study, in addition to off-odor analysis, we can use GCMS for general flavor profiling, which allows us to compare between samples for whatever reason the client has. So this could include determining the impact of different processing or ingredient changes or storage conditions on the overall flavor profile of a product. So I'm going to show an example here where a client was developing a broth product, and they were trying to use a cured meat to create a broth that would have similar aromatic properties, but they weren't satisfied with how that broth was coming out. So we compare the two samples, and you can see they both have very complex profiles here. We have the cured meat on the bottom in black and the broth on top in red. And I'm just going to highlight a few here. These compounds shown with orange arrows, these are all sweetness compounds, which are showing up in our cured meat, but not showing up abundantly in our broth. And then in blue, these compounds are acidic or fatty, which you would expect for a broth. And then finally, these compounds in green, these are pyrazines, which Jamie had mentioned earlier. These are found in many different roasted products, including coffees and cured meats, and they contribute roasted nutty aromas, so they can be very important in many different types of products. So with this analysis, we were able to find that these two samples did share a number of major volatiles, such as acetic acid, but the cured meat also contained many compounds that are associated with sweet, buttery aromas and also roasted odors, while the broth had much higher intensities of fatty and acidic compounds. So with this information, the client was able to further pursue how to more accurately get those aromatic flavors into their final product. So there are additional techniques that we can include when we want to further our GCMS analysis and support our results. We can apply GCMS in a unique way using MSMS or tandem mass spectroscopy. And this is a targeted technique where the mass spec is set to actually filter out specific ions and fragments. And this allows us to have a very high level of sensitivity and selectivity. So if we identified a unique compound of interest based on our screening techniques, we could then look into low-level quantitation or detection using GCMSMS. And then additionally, there are many other characteristics that we can look at for off-odor analysis, including, aside from GCMS. These can include looking at metals or heavy metals content using ICP-OES or ICP-MS. We can also look at microbial analysis, where we can investigate the types of species that are present and the general amounts to look for differences between samples. We can also involve sensory panelists who can give an expert opinion on the properties of the samples. And we can look at acidity, alkalinity, and peroxide, which are all related to those sample properties. And then finally, we can look at non-volatiles using liquid chromatography. And that dye screen is just one example of a way that we could use liquid chromatography to further our investigation into the sample properties. So in summary, SPME-GC-MS is a powerful technique for off-odor analysis and volatile profiling. And by comparing the profile of a control and suspect sample, we can identify compounds that could be responsible for the off-odor. And then we can use these identifications to correct or find the underlying issue. So thank you very much for listening today, and we'd be happy to take questions at this time.
Thank you for the presentation, Jamie and Lily. Now we will move on to a short Q&A. Feel free to continue to submit questions during this session. If we do not address your question, you will be asked live during the presentation. All right, we do have one question here. Our first question is, can GCMS techniques be applied to products such as pet foods?
Yep. Okay, I can take that one. Yes, this approach can be applied to pet food. As mentioned earlier, we have techniques such as SPME that allow us to apply this to very complex matrices. But we can even apply it to products that a human isn't going to be eating, such as pet foods, by comparing a control and a suspect sample. We've also applied this method to products such as cosmetics and even packaging materials, as Lily mentioned.
All right, thank you. Our next question here is, how do you know if a difference in profiles is significant?
Sure. So we have several strategies we use when we are looking at our data at the end of this process. We have to consider, based on the product, what compounds would we expect to see, and would we think that would be naturally occurring. For example, going back to our fruity beverage, seeing something like isopropyl alcohol was not something we would expect to find. And then in addition, we look at how significant is the difference. So if we have a very large peak that is only in one sample, that is likely to be significant. And then we also look at, is this compound actually likely to impact flavor and odor? So we have to consider how these differences could actually be linked to an underlying cause.
All right, thank you. Our next question here is there a comprehensive article or reference that ties molecules into their associated flavor/odor?
There are several databases that we do use for this. One of them is from the Good Sense company. It has a very extensive compilation of specific properties that are associated with specific compounds. So yes, we do apply many different techniques to try to investigate, but it's true that sometimes we see differences in compounds that don't have a known aroma, and then in that case, it can be difficult to know if that is actually linked to the off-odor or not.
All right, thank you. Our next question here is, are you able to perform analysis if you don't have a control sample?
I can take that one. Yes, we can. It's not ideal, as you probably saw on a bunch of our slides. Profiles of food and related products tend to be very complex. We can look at products without a control and look for anything that might be an obvious source of off-odor, such as a solvent or like a fragrance that is associated more with, say, soaps and cleaning agents. However, if we're looking at, say, a subtle difference in profile that might be due to like an ingredient issue, a control is ideal.
Thank you. Our next question here is, does Eurofins ever couple sensory analysis with GCMS analysis to identify off flavors, off odors, or unusual mouthfeel in foods and beverages?
So yes, we have used sensory panels. We do have options to send samples to trained sensory panels where they can compare the control and the suspect. Say, for instance, you've received a complaint, but you want to confirm, in fact, that there is an off-odor and off-flavor. And so we can use industry-recognized methods to characterize the flavor and aroma, as well as any mouthfeel or texture issues.
Thank you. Our next question here is, in the event that microbiological degradation is the cause of off-odor, off-flavor in food, can you identify the offending or microorganism or narrow down the classification of microorganism that is causing the problem?
Sure. So in some cases, it is possible to isolate microorganisms that may be causing the off flavors and off odors. Typically, we would culture both the control and the suspect on a variety of media with various conditions. And then we would look at the dominant colony forming units, or CFUs. And then these can be compared further by looking at metagenomics sequencing and comparing that to a database to identify the actual specific microorganisms that are present. And in this case, very similar to our other analyses, it is important to have a control because we would have to look for, once again, differences in the microbes that are present in order to actually identify if there's one that should not be there.
All right, thank you. And then our second to last question here is, just curious, a lot of people are concerned about water bottles sitting in your car in the summer. Have you done any tests on this, and what did you conclude?
Well, we haven't done that specifically. We have done contaminated water bottles where they've been contaminated by factors like cigarette smoke and environmental conditions. And there we could, in fact, see evidence of the contamination.
Right, and I think just this is, Josh, to add to that, really, companies before they use these bottles do extensive leaching studies. So if there was a particular complaint, we certainly could, but haven't had any specific investigations in that regard.
Thank you both. Our next question here is, what kind of compounds can be detected in pet foods?
I can take that one too. So, really our techniques aren't limited to the types of compounds we can detect. When I think of a pet food in particular, I would be looking for rancid compounds, potentially volatile aldehydes, or really it would depend on the description of the off flavor or off odor. But we're not limited. Anything that's volatile can be detected.
All right, thank you, Josh. Okay, that wraps up our Q&A session for today. I want to remind everyone that today you attended part 1 of a two-part webinar series. Part 2, Foreign Materials and Unknown Objects, will be on August 17th from 11 a.m. to 12 p.m. Central Time. The link to register is going to be located in the chat below. Also, a recording of this webinar along with the slides will be available in 3 business days. Thank you for your expertise and presentation today. Thanks everyone for attending.